So this is the the part of the meeting that I think we're all excited to see, which is an important part of our regular meeting, which we're bringing back this year. So so we'll have three abstracts presented, which were ranked as the best abstracts in a more clinical sort of arena, and those are nominated for the Nesbitt Award, and then followed by three that are more basic in terms of their scientific aspect, and those are nominated for the Nesoloff Award. So without any further ado, let's see here. So let's switch over. So our first speaker will be Dmitry Evseev, if I pronounced that correctly. Hopefully I did. And so we have Dmitry there, and I believe you should be, oh, sorry, I said that backwards, excuse me. I actually, Kathy just reminded me that Nesoloff goes first, even though our program has it listed the other way around. That's okay. So, so instead, actually, we'll start with Emily Landy. So we'll start with the basic scientific abstract. So Emily Landy will be presenting an abstract entitled Cytotoxic Impairment and Excess IL-18 Drives Spontaneous Oligoclonal CD8 T Cell Hyperactivation and Hyperinflammatory Disease. So do we have Emily? Can she come right there? You are. Thank you. Yes. Now, can you share your screen?
Let's see.
You should have permission to do that. All right. Yes, we see it. We hear you. It's all great. So please proceed.
All right. Well, thank you everybody for showing up, and thanks for the opportunity to talk about the stuff that we're working on in the Cano Lab. So our lab works on understanding the mechanisms behind hyperinflammation. So obviously I don't need to tell you what hyperinflammation is, but here is just a quick summary of the clinical phenotype that we see in the patients. And as Jana pointed out, typically this is triggered by infection. However, not every single person who gets an infection actually ends up in the hospital with hyperinflammation. So we're interested in what makes a person susceptible to this. And one such mechanism is, again, abnormal cytotoxicity. So just a quick overview, normally when an NK or CD8 T cell comes into contact with an appropriately activated target cell, it will release perforin-containing granzymes into the immune synapse, thus killing this target cell and further terminating any more immune response that goes on. However, if a person has a defect in cytotoxicity, say perforin deficiency, their NKs and CD8s can't actually properly terminate this immune synapse, and we end up with what we all know as familial HLH. Now, on the flip side, hyperinflammation in patients with rheumatic diseases goes by the name of macrophage activation syndrome, or MAS. And this is characterized by extremely high levels of serum IL-18. And in this case, the serum IL-18 levels actually completely overwhelm the endogenous inhibitor protein, IL-18 binding protein. And this allows for free IL-18 to then signal on cytotoxic T cells. And so excess 18 appears to be driving MAS in a number of diseases. And this meta-analysis summarizes what we've known for some time, which is that very high levels of IL-18 can be used diagnostically to separate systemic JIA and JIA MAS from nearly every other rheumatic, infectious, malignant, inflammatory disease, and that's including HLH. But IL-18 is more than a biomarker. There are now 3 monogenic diseases that are associated with chronically high levels of IL-18, and this really puts IL-18 upstream of HLH and MAS in these patients. And so in order to study the effects of chronic IL-18, we use IL-18 transgenic mice, or 18 transgenic mice, and these are mice that constitutively produce excessive amounts of mature IL-18. And then we also use perforin knockout mice, which are essentially a hyperinflammatory control, because we know if we infect these mice with LCMV, they actually can't clear the virus and they end up with persistent viremia and unrestricted antigen presentation. So we hypothesized here that the IL-18 transgenic mice actually wouldn't have any hyperinflammatory response. However, to our surprise, the IL-18 transgenic mice actually ended up with almost as severe of immunopathology as these perforin knockout mice. And while the degree and the kinetics of the systemic interferon gamma production were similar to these perforin knockout mice, The IL-18 transgenic mice actually clear virus just as well as a wild type mouse. And from published data that I don't have the time to show everything to you, obviously, we know that this LCMV-induced hyperinflammation relies on interferon gamma and CD8 T cells. And importantly, also, this is through an IL-18 mediated phenotype. So when we actually remove the IL-18 receptor on all the T cells in a IL-18 transgenic mouse and give it LCMV, those mice are actually protected from this LCMV-induced hyperinflammation that we would normally see. And so when we looked at the CD8 T cells in these mice, we see that in the perforin knockout and the IL-18 transgenic mice, that they have this hyperactivated phenotype. And this phenotype is characterized by an increased expression of inhibitory receptors. And these are the receptors that are most commonly associated with exhausted T cells. However, unlike fully exhausted T cells, our CD8 T cells are actually producing a whole lot of pro-inflammatory cytokines. And so this together says that, like, while the clearance of LCMV in these 18 transgenic mice are just normal, we're still getting this hyperactivated CD8 phenotype, providing evidence that indeed excess IL-18 on its own can actually lead to at least infection-induced hyperinflammation. But that's not to say that these susceptibility factors, meaning cytotoxic impairment and high IL-18, actually work independently all the time. In fact, it's actually been shown that in patients with MAS, those patients who have high IL-18 all the time actually have an enrichment for partial cytotoxic defects. And this is despite the fact that there's no apparent disadvantage of having partial defects in cytotoxicity just on its own. And so this led us to wonder how excess IL-18 and cytotoxic impairment might actually be interacting. So we crossed these two mice to make mice that both lacked perforin and had excess IL-18 all the time in order to study them during an LCMV-induced hyperinflammation setting. However, they ended up getting hyperinflammation before we could even infect them. And this is especially true in these perforin knockout IL-18 transgenic mice. By about 6 weeks, these mice end up with severe spontaneous hyperinflammation, and it's often lethal. And even in the mice that most represent those patients, so the perforin heterozygous IL-18 transgenic mice, they have this intermediate hyperinflammatory phenotype, as shown here by the systemic interferon gamma levels, as well as anemia. And I'll point out, again, this is in a homeostatic state. And so when we looked at the splenic cells in these mice, we see that there's this population of CD8 T cells that pops up that's very prominent in these dual susceptibility containing mice. And they seem to be hyperactivated. So they are, again, expressing high levels of the pro-inflammatory side, or inhibitory receptors, but they are producing basically all the pro-inflammatory cytokines like gangbusters. And so what we were really interested at this point was what's actually driving this big population of CD8 T cells in these mice, which are not being infected with anything. And so we utilized TCR-seq, so we did interferon gamma depletion because we know interferon gamma is important in this, and they do get better. However, since the CD8s are probably the cells that actually are producing all of this gamma, we wanted to look at the CD8s. So when we look at the clonality of the CD8s in these, again, homeostatic conditions of all four of these genotypes, we see that the CD8 T cells have this hyperexpansion of the clones, meaning that this is some sort of clonal expansion going on in the CD8s in these mice. And so from here, we hypothesized that, again, these hyperexpanded, hyperactivated CD8 T cells that we're seeing homeostatically in the dual susceptibility mice might be to some sort of maybe low-lying infection or something like that. So we compared the TCR clones from all the hyperexpanded clones in all of these mice. And I'm just showing you one for example. But they don't show up to any appreciable degree in any of these other mice, suggesting that this may be stochastic or spontaneous in nature and not necessarily from a specific antigen or an infection. And so at the same time, we wanted to know what these hyperactivated CD8 T cells were doing transcriptionally. So again, we took these CD8 T cells from the homeostatic conditions in all 4 mice. And what you can see here is that the perforin knockout IL-18 transgenic CD8s are sequestering away from this main group of CD8 T cells, whereas the perforin heterozygous here in the pink IL-18 transgenic and the IL-18 transgenic alone are sequestered away from what we would typically see in the perforin knockout mice, which are essentially fine until we infect them. And when we look at the genes that are driving this variant in this huge PC1, we see that again, these CD8 T cells in the perforin knockout IL-18 transgenic mice are actually showing a hyperactivated phenotype. So they have this increased expression of the inhibitory receptors, as well as an increased expression of these effector genes, such as interferon gamma and granzyme. Whereas on the other side, with these perforin knockouts, we see more of a central memory phenotype. And so in light of this transcriptional data, we hypothesized that maybe these CD8 T cells in the perforin knockout IL-18 transgenic mice are actually on a path towards an exhausted phenotype. And so we chose to compare our data set to one that had both a precursor and a terminally exhausted pool of cells. And so this Haining data set was perfect for that, because essentially it gives us a kind of a timeline of sorts in the world of CD8 T cell exhaustion. And so within this data set alone, you can see clearly that we can show this transition from an exhaustion precursor to a terminally exhausted CD8 T cell. And this is both in an LCMV clone 13 as well as tumor setting. And then when we overlay our data, or when we compare our data within this, you can see that we see the same type of transition where the IL-18 transgenics and the perforin knockouts are more in line with a progenitor cell for exhaustion, whereas you see this transition into a perforin knockout IL-18 transgenic, which is again, more like this terminal exhausted transcriptome. However, there is a big difference in these, is that these terminally exhausted CD8s actually are not producing and releasing pro-inflammatory cytokines like our IL-18 transgenic perforin knockout mice do. And so at this point, just a quick summary of what I've gone through is I've shown you that both high IL-18 as well as cytotoxic impairment can be independent and synergistic in causing hyperinflammation. And this is both in mice and humans. And I've also given you an example of how a heterozygous mutation in perforin or cytotoxicity can lead to hyperinflammation in the context of high IL-18. And then from the mouse data, I've shown you that hyperactivated CD8 T cells are the main effector cell type in both infectious mediated as well as the spontaneous HLH. And in particular, those hyperactivated CD8 T cells that we're seeing homeostatically in those dual susceptibility mice have a transcriptome that is more of a terminal effector in terms of the pro-inflammatory cytokine production, but also have all of these inhibitory receptor, just like in terminally exhausted CD8. And then on top of that, these CD8 T cells are stochastic and clonal in expansion. And so this really suggests that the CD8s that we're seeing here are being proliferated in terms of antigen. So this is an antigen-dependent mechanism. And right now we're actively trying to determine the nature of this antigen. But altogether, I hope this really told you that we should be strongly taking into account the environmental triggers, but also these multiple susceptibility pathways that are potentially involved whenever we're treating patients with hyperinflammatory diseases. And so with that, I'd like to thank everyone for their attention. And I thank the people and the funding that made this possible, and I'll take any questions.
All right, excellent. Thank you, Emily. Again, if you have a question, just use the raise hand feature. I like the can of cytokine storm. That's that's awesome. So I had a couple questions, and one is that when you're looking at the clones, I assume you're looking in the spleens of the mice.
Yes, this is spleen.
Yeah, I mean, the obvious candidate, of course, is that they're expanding in response to, say, microbial antigens. So like in the gut. Have you looked in notobiotic mice Or have you tried looking in clones in the gut, for instance, for comparison?
So we haven't been able to do this in like a germ-free situation, but that's something that we're definitely interested in looking into.
Mhm. Got it. All right. And then also the gene expression comparisons, these were all in sort of uninfected mice, right? In other words, these were all sort of at rest. So
Mhm.
really you're comparing these activated cells against essentially non-activated cells. Wouldn't a more fair comparison be against, say animals infected with virus? If, if
Sure.
it's possible to do that?
Sure, yeah. So maybe I have data that I just didn't have time to show you guys, but I did compare this data set to actually we have transgenic IL-18 transgenics and peripheral knockouts that are infected. And so we see the same sort of idea where the peripheral knockout IL-18 transgenics do look more like those terminal effectors that we would see in a wild type or an IL-18 transgenic during LCMV infection.
Mm-hmm. Excellent. All right. Well, thank you very much. I don't see any raised hands, and our time is up, so we should move on. Thank you, Emily. That was a fascinating presentation. So next up is Paul Milne, who will be speaking about lineage switch of cells harboring BRAF V600E alleles in patients with high-risk LCH treated with inhibitors. And I see we have Paul right here, and I see his slide. So why don't you take it away from here, please?
So yeah, thanks, Michael, and thanks for the committee for picking this presentation today. You might have to bear with me. I've got a wee bit of a sore throat that I must have picked up last night at the dance party, but I'll do my best. So this study was a longitudinal study looking at V600E alleles in patients over their treatment time course when they're on inhibitors. So we know that the patients have 60% of LCH patients have V600E mutation in their lesions. And in my work and in other labs have shown that the V600E can be found in the bone marrow and in the blood. So this is a diagram of hematopoiesis we use now, which is fairly dynamic. Everything starts off as hematopoietic stem cell in the bone marrow. And then there's a gradual decrease in potential as the precursors lead on to the mature cell types. So in the bone marrow, we've seen that the majority of V600E is enriched in the hematopoietic stem cells and the various myeloid progenitors, while in the blood, it's enriched in the monocytes and the myeloid DC subsets. So this leads on to the idea that these cell types could be the precursors of LCH. We know that patients with high-risk LCH who progress through frontline therapy have conventionally been treated with chemotherapy, which works quite well, but if they fail, where we see reductions in circulating V600E alleles. You can see the reductions here in PBMC, which is the steady line, and the plasma, which is the dashed line. Inhibitors of BRAF, such as vemurafenib, can also induce a dramatic clinical response on those patients who fail the salvage. But in these patients, we can see static levels of V600E mutation throughout treatment. So this leads on to the question of whether the clonal architecture of V600E mutated cells within the circulation changes throughout the treatment time course. So for this study, we included basically any patients who had undergone chemotherapy or MAP kinase pathway inhibition, where we had multiple samples to look at over a number of years. So we included 6 patients, 2 of which had chemotherapy only, 4 patients who had MAP kinase pathway inhibition plus minus chemotherapy. This included 2 patients on vemurafenib, 1 on dabrafenib, and 1 on trametinib. This slide is just going to show the workflow of the experiment. So we take the EDTA blood samples. From here, we can take a true count, which is a flow cytometry tube that has a very exact number of beads inside. You can run this through the cytometer with the markers of interest, and you can derive an absolute cell count per microliter of the various subsets you want to look at. You then could take neat plasma and PBMC and test these for the V600E mutation, and in our lab we use a TACMAN mutation detection system, and this gives you the mutated cell fraction. We then sort out the different cell fractions from the blood, from the PBMC, using flow cytometry, and again test each fraction for the mutation. So then when you do a simple calculation, you can derive the mutated cells per microliter for each cell type. So I don't have much time to go over the gating strategy we use, but we've used some tips from Florent Junot's lab, such as using CD88 to clean up the myeloid cells. And we collected the CD14 positive classical monocytes, CD16 positive non-classical monocytes, and also the myeloid dendritic cells, which encompasses the DC2s and DC3s, which we didn't split just to limiting cell numbers. And we also collected T cells just as a control. So looking at the different patients, this first patient was a 7-month-old female with risk organ positive multisystem LCH who failed the initial chemotherapy, had a good response on salvage chemotherapy. This just shows the disease activity score. So as you can see, there was a gradual decline in circulating mutated alleles in the PBMC, again in the steady line and plasma of the dashed line throughout the treatment. A very similar pattern is seen in the sorted cell subsets with positivity in the monocytes, which is red and orange here, and in the DCs, which is in blue. And then determine the absolute number of circulating V600A positive alleles of each subset, and then display these data as pie graphs. And this shows the disease burden for each cell type at each time point. So we can see that the monocytes contributed the majority of the disease burden in this patient, and that these cells were ablated with conventional chemotherapy. So this patient remains in clinical remission with undetectable V600E 2 years after diagnosis. Second patient is a 3-year-old girl with a diagnosis of risk organ negative multisystem LCH. She didn't respond to intermediate or escalated chemotherapy, but she responded to the salvage chemotherapy. The lesions were resolved, and the lower levels of V600E mutation that we'd seen in the circulation were ablated. Again, the subset testing revealed very similar results to the first patient with mutated alleles, predominantly in the monocytes and then the dendritic cells, which cleared during the therapy. So going on to the patients who underwent inhibitor therapy, this is a 5-month-old girl with risk organ positive multisystem LCH. She initially responded to the chemotherapy, had a disease recurrence at 5 months with a disease activity score of 10. She was then given vemurafenib, which is shown in the red bars, and had a really rapid disease control. However, although her disease rapidly regressed on the introduction of vemurafenib, there was high levels of the mutated alleles throughout the treatment in both the PBMC and plasma. And this is consistent with previous reports from the labs. Surprisingly, though, when we looked at the different cell subsets we'd sorted, we can see increasing mutated levels of mutated DNA in the T cells, which is this black line here, throughout the treatment. In fact, after 4 years of treatment with vemurafenib, the mutated DNA was only seen in the T cells. There was no mutated DNA in myeloid cells whatsoever. So 3 further patients who were refractory to chemotherapy went on to receive inhibitors. They all maintained a high level of V600E in cellular and cell-free DNA for several years. Fractionation of the mononuclear cells during this treatment time courses of these patients again revealed this surprising shift of mutated alleles to the T cell compartment. So we were surprised to see this positivity in T cells. We were really only using these as a negative control at the start of the study. But now we've seen this positivity in the T cells, we really needed to drive into all of the different lymphoid fractions into the T cell subsets and also in the B and NK cells. So for this, we used a different sorting strategy. First of all, taking the CD4 positive T cells, splitting them by CD4 and 8. Take 45RA positive naive T cells, 45RA negative central memory T cells, 27 negative effector memory T cells, and TEMRA cells from the CD8 positive fraction. We also took NK cells and B cells, and the classical monocytes, non-classical monocytes, and DCs as shown before. So for this part of the study, we looked at 3 different patients who had undergone inhibitor therapy, one with VEM, one with DAB, and one with trametinib, and selected the time points where there was a predominance of mutation within the T cells. So these first pie graphs just show that there was no mutation in the myeloid fractions as we've seen previously, and all of the mutation was seen in total lymphoid fractions. When we drove down into the lymphoid cells, you could see that the majority of the burden of the mutation is in the CD4 positive T cells, which are in orange, and the CD8 positive T cells in blue. Again, driving down further into the different T cell subsets, this is where we didn't particularly see a pattern emerging just in these 3 patients. But the fact that there was a high level of mutated DNA in the naive T cells is further indication that this is hematopoietic stem cell disease. So just concluding, we can see that V600E was clear during chemotherapy, just with the general ablation of the entire hematopoiesis. However, inhibitors fail to clear mutated DNA. In fact, there's a shift in the DNA to the lymphoid cell compartments, and that these mutated alleles in the BNK naive cell subsets is consistent with the hematopoietic stem cell origin. So the big question now is whether we can use this information in the clinic. So we know that inhibitor withdrawal is rarely successful within the first couple of years of treatment. This first patient, the clinicians tried to withdraw inhibitor therapy, and both times they did this, there was a rapid recurrence of disease. However, when we look at these 2 other patients on inhibitor therapy, we can see that they were taken off the inhibitor and there was no systemic relapse. And at the time points that the patients were taken off the inhibitor, there was no circulating myeloid V600E positive DNA. So this brings the question of whether this transition from a myeloid to lymphoid cell reservoir is what we need to look for to mark the potential for withdrawal of the targeted therapy. However, this isn't the full story, and we need to be really careful with the patients. One of our patients had a diagnosis of neurodegeneration a year after they were taken off the vemurafenib therapy. This first image just shows that there was a relatively normal MRI during the time that the patient was taking vemurafenib therapy. And at this time point, the mutational burden was almost exclusively in the classical monocytes. However, in images B and C, we can see there's progressive signal changes in the cerebellum and cavitation in the dentate nucleus indicated by those orange arrows, as well as a mild global atrophy of the cerebral hemisphere with singular changes in the basal ganglia structures indicated by the blue and yellow arrows. At these later time points, the patient was no longer on vemurafenib, but the mutated DNA was only present in the T cells. We do know that T cells can recirculate into the brain in the steady state, and there's been reviews of LCH showing that there's a large T cell infiltrate in the brain of neurodegenerative LCH patients. So we really need to look at now how mutated T cells play a role in neurodegeneration in LCH. So with that, I'd just like to thank everyone in our lab in the UK, all of the patients and their families for all of the samples that they've sent, as well as the clinicians for taking those samples, and all of the funders for the research. Thank you.
All right, excellent. Thank you, Paul. You guys all look busy. You're drinking coffee, huh? So, all right. So we have a couple moments for questions. I see that Astrid has her hand raised. So Astrid, do you want to go ahead and ask your question?
Yeah, sure. Thanks, Mike. Thank you, Paul, for this very comprehensive overview of, I think, quite recent data that you have been collecting in Newcastle. It's very nice to see that at least you guys in Newcastle also confirm what we earlier published, that the lymphocytes are probably as often affected by the mutation as the myeloid cells, but somehow in most patients, the myeloid compartment sort of pops out, whereas it's a little bit more difficult to find the mutated lymphocytes. My question to you is, do you really think that this change from myeloid to lymphoid is a general phenomenon? Because I'm sure we have seen patients that don't show this pattern, and I'm also pretty confident that Caroline also has patients that don't show this real switch. So what is your comment on this?
I mean, we're looking at a small amount of patients, but we have followed them over a long time period, over 5 to 6 years. So I think the switch is just incredibly gradual on the inhibitor, and it'll depend on how strong the treatment is given by the clinicians, I imagine, to how quickly this switch occurs.
Can you somehow relate it to the pharmacological levels of the inhibitor?
Exactly. So that's what I would imagine. And so obviously, if there's more inhibitor, I would imagine that the stress on the cells to shift down the lymphoid pathway would be greater. So quite a lot of these patients are quite young, and the inhibitor strengths are quite low. So I imagine it would take quite a long time.
All right. Okay, good. Thank you. I see Ashish also has his hand raised. Ashish, if you could ask your question really quickly, then we could do that one before we move on.
Great. Paul, just two questions for you to consider and more of speculation. Number one, could this just be the natural history of this disorder? Because many of these patients before inhibitors probably would not have survived. So is it just that we're seeing the natural evolution? That is question number one. And the question number two, in the patient who developed neurodegenerative disease upon withdrawal, I know it coincided with the appearance of the mutation in the lymphocytes, but we do know that the MRI appearance lags behind the actual event. So is it possible that what you're seeing was damage that was caused by the myeloid cells? Again, it's pure speculation. And then what you're seeing in the MRI is the fibrosis that follows. It happens to coincide with when the cells appeared only in the lymphocyte compartment. It's more of speculative questions just to see what you thought about that.
I mean, I would have to give speculative answers back. So we know the brain lesions are completely different to any other lesions elsewhere in the body. So there's a good possibility that those particular lesions are caused by a different cell type. I believe there's no Langerhans positive 1A positive cells in the brain lesions. There's a lot of different types of damage to the basal ganglia, etc.
All right. Okay. Thank you very much, Paul. Thank you very much, Paul. That was fascinating. There are more questions in the chat if you want to carry on with those. So we'll move on to our next presentation, or next presenter, excuse me, who's Danielle Menachino, who is going to present a talk entitled BRAF V600E expression in dendritic cells reprograms translational dynamics to increase LPS-induced TNF-alpha production. So I see Danielle's slide. There you are, Danielle. I see you as well. All right. Okay, take it away.
You can hear me fine?
Yes, great. Thank you.
Great. Thanks for that introduction, Mike. So what I'm going to be talking about today is really a summary of the LCH project that I started in Ed's Lab a few years ago. And all of the data that you're going to see today is unpublished, but currently getting drafted into a manuscript, and hopefully you'll see it published soon. So we know that much work has gone into defining the current pathogenic model for LCH, which is that some precursor cell acquires a somatic gain-of-function mutation in the MAP-ERK pathway, and this is commonly BRAF V600E. This drives the aberrant differentiation into a dendritic cell-like cell, which I'll be referring to as LCH cells, which then seed different organs and drive the inflammatory lesions. And so to better understand how these cells lead to disease, many researchers have focused on this MAP-ERK pathway, which we know connects environmental stimuli to a myriad of intracellular processes. And many labs, some that are tuned in today, have done a lot of work on understanding how some of these processes are affected in a hyper-ERK environment. And what our lab would like to do is contribute a better understanding of how the inflammatory functions of these dendritic cells are affected. DCs are dynamic, reactive inflammatory cells that specialize in sensing pathogens through specialized receptors, including toll-like receptors, or TLRs, that signal through ERK to then orchestrate the adaptive immune response. Given our lab's expertise in TLR signaling and the fact that TLR signal through this pathway, we decided to investigate how these cells react to TLR4 stimulation, with particular interest in the release of pro-inflammatory cytokine TNF-alpha. Under the hypothesis that V600E expression disrupts the TLR-induced cytokine responses in these cells. And we predict that there would be an amplified signal downstream of ERK, which would lead to increased production and secretion of TNF-alpha. To answer this question, we use one of the established LCH mouse models, which expresses a single BRAF V600E allele under the control of CD11C promoter, which is a DC specific marker. So about all of the dendritic cells in this mouse express V600E, and they get a very severe form of LCH spontaneously. To gauge their response to TLR4 stimulation, I injected wild type and LCH mice with LPS and measured circulating TNF-alpha 2 hours later. And as you can see here in figure B, there's a 400-fold increase in serum TNF-alpha in the LCH mice in red compared to the wild type in black. And that color scheme of LCH being red and wild type being black is going to remain consistent throughout this talk. To better get a direct reading on what the dendritic cells in this mouse are contributing, I generated bone marrow derived dendritic cells, or BMDCs, and I confirmed that there's an increase in levels of phosphorylated ERK in the V600E BMDCs from the LCH mice compared to the wild type. And what's really cool is that I can pre-treat these cells with a specific V600E inhibitor, one of the paradox breakers, and bring that level of phospho-ERK down. Using this system, I treat cells with LPS and measure TNF-alpha secretion over time. And I've shown here that there is a consistent increase in the secreted TNF-alpha, which is almost completely reversed with the inhibitor. And I show summary data from an F. Now, fairly routinely, I checked the transcription levels of TNF-alpha from both splenocytes from these mice and the BMDCs at the 4-hour time point. And now we see that although there is an increase in the levels of transcripts at baseline, to our surprise, there is a decrease in LPS-induced TNF-alpha transcripts in the LCH mice compared to the wild type, both in the splenocytes and in the BMDC model. And this is not reversible. And focusing on the BMDC plot, I've done this in 5 separate experiments. And so each dot here is showing you the average of 3 biological replicates for each experiment. And so this data is suggesting that there is a increase in TNF-alpha protein despite there being a decrease in transcription. And what I'm not showing here is that I have also looked at NF-kappa B signaling and TLR4 expression, which are both down. So this kind of made me wonder whether or not these cells are actually producing more protein on a per cell basis. And to get an accurate reading of that, I did some intracellular flow cytometry. So this plot is showing us in our LPS stimulated BMDCs that there's a uniform increase in TNF-alpha protein on a per cell basis in the LCH BMDCs compared to the wild type. And the dotted line here indicates the V600E inhibited samples, which tells us that it is partially reversible. And I've summarized this data from 2 independent experiments on the right. So this does tell us that there is an increase in TNF-alpha protein being produced, despite there being a decrease in signaling and TLR4 transcription. To understand the mechanism behind this, we considered all of the ERK-driven post-transcriptional mechanisms that could possibly lead to an increase in production and or secretion of TNF-alpha in these cells in a hyper-ERK environment. And although negative data is extremely important, for the sake of time in this presentation, I won't be showing it. So for now, you'll have to take my word that I have measured the reuptake of TNF-alpha in these cells and shown that it is unaffected, as well as the rate of degradation of TNF-alpha, which is also undisturbed. And we'll start with cleavage of TNF-alpha, which is a TNF-specific mechanism. Since the secretion of membrane-bound TNF-alpha depends on the enzymatic cleavage by TACE, we can determine the rate of secretion by measuring TACE activity. Using a fluorescent-based in vitro assay, I determined that there is a significant increase in TACE activity in the LCH cells, which is acutely reversible. So we know that whatever TNF is being produced is getting cleaved and thus secreted at a faster rate in the LCH BMDCs. Now moving on to translation, it's a little bit more complicated. We can infer the rate of translation by determining the number of ribosomes working at any given point on a transcript. So if there's one ribosome or a monosome, it's indicative of rather slow translation compared to transcripts that have multiple ribosomes or polysomes working on them at any given point. To measure this, we fractionate cell lysates over sucrose gradient to separate the polysomes. And then we can measure the concentration of RNA in each fraction. And this representative plot on the left, or sorry, on the right, shows us a very impressive increase in the number of transcripts bound by polysomes in the LCH samples in red. This indicates a very strong increase in translation compared to the wild type. Again, the dotted line indicates the inhibited cells, and this plot tells us that this phenotype is not acutely reversible. I've also plotted the bound ratio of the bound versus unbound for 2 independent experiments. And to ensure that TNF alpha is one of these transcripts bound by the polysomes, I've isolated RNA and done some qPCR to measure the translational efficiency and shown that TNF alpha translation is up in particular. Now, so we know that there are more ribosomes bound to the transcripts from this data, but what we'd like to know is if they're moving at a faster speed. So to measure elongation, we can use this pyrimycin-based flow cytometry assay that tells us, it determines the rate of ribosomal runoff over time. So these representative plots on the left show the time it takes for ribosomes to drop off the transcripts, indicating their speed or elongation. So you can see that the ribosomal signal for V600E cells decreases at a faster rate, indicating an increase in elongation. And I've plotted the nonlinear regression of the MFIs from 2 independent experiments on the right, showing a significant difference between the genotypes. Additionally, I've looked at one of the main elongation factors, EEF2, which regulates elongation and is downstream of ERK. So when ERK is activated, it actually prevents the phosphorylation of EEF2, which then actually allows for elongation to happen. Phosphorylated EEF2 actually slows down elongation. And so we'd expect our V600E cells to have less phospho-EEF2, which is what we see by Western blot. So there's less phospho-EEF2, and that actually gets replenished with the inhibitor. So this is a reversible phenotype, and that is summarized from 2 independent experiments below. So taken together, this data tells us that V600E expression increases the rate of translation. And in sum, I've shown you that V600E expression in these cells influences them in a way to allow them to produce and secrete increased TNF alpha despite there being less transcription and signal. And in understanding this mechanism, we've identified 2 ERK-driven post-transcriptional mechanisms, one TNF specific, which is the cleavage bitase, and one more global, which is the rate of translation. Now, we're not the first to demonstrate that V600E leads to an increase in translation. For example, it's been shown in melanoma cells, but I think the important distinction here is that the consequence of that translation is specific for immune cells rather than a melanocyte. And a melanocyte, this could lead to increased proliferation, and we know that's not the case for our dendritic cells, and it's probably increased inflammation. And taking another step back, I think the moral of this story, which can apply to many other experiments and research, is that RNA is not the bottom line, and it should be complemented with proteomics as well. Next steps for us include understanding the global impacts of the increase in translation using RNA-seq from our polysomes to define the translatome. And while I think a lot more work needs to be done, this project opens the door to targeting translation as a potential therapy. And I would like to thank Ed's lab, both current and previous members, my committee, Shang and the Wei Tong Lab, who did all the polysome work. And of course, a big thank you to the Histiocyte Society, the organizers and the coordinators, and a thank you for this nomination. And with that, I will take any questions.
Okay, excellent. Thank you, Danielle. That was a very impressive sort of dissection of some of the cell biology here. I'm very, polysomes, it's basically, seems to me it's almost impossible to make them look so good. So, but so anyhow, so again, if there are any questions, just use the raise hand feature in the chat and we can call on you. One of the things I'm wondering about is, is it known, do myeloid cells, do they have sort of, stores of mRNA that are not being translated? There's a phenomenon like that in T cells, but I'm not aware of that in myeloid cells where, you have sort of a bunch of message sort of in suspended kind of waiting, right, for rapid translation, for instance, like in T cells with antigenic activation. Is there anything like that in myeloid cells? Because I always assumed that was not the case.
Yeah, I think
receptors.
I think it's been shown in macrophages, and I'm not sure in dendritic cells that they do also have a store of specifically TNF, but I didn't see any increase in like stores of TNF RNA at baseline.
Yeah. So, all right. Well, it'd be interesting to see what else is affected besides inflammatory mediators in these cells, right? If this is a global MAP kinase impact on these cells, that could explain a lot of things in terms of abnormal biology.
Yeah, and I think Paul's the presentation before this, right? It's highlighting that not only dendritic cells should be looked into, but all of these other inflammatory cells that have these specialized inflammatory functions and possibly expressing V600E. It brings up a lot of really interesting questions.
Yeah, indeed. All right, excellent. Well, thank you very much, Danielle.
Thanks,
You're very welcome. All right, so let's move on to our next abstract. So those first three ones were the basic science ones, and next are the more clinically oriented ones. And so this the next presenter is Dmitry Evseev, and Dmitry will be presenting the interim results of a multi-center study of safety and efficacy of vemurafenib and cytarabine plus cladribine therapy in high-risk pediatric Langerhans cell histiocytosis with BRAF V600E mutations. So do we have Dmitry on the platform? I see.
Just a second. Yeah, I'm here, but I've got some problems with screen sharing.
Okay. Is it giving you permission?
No, I think. Well, I have the permission, but I've got some certain. Okay, I'll try to do it.
You should have it now.
Yeah. Thank you. I'll try to do that.
So we hear you well, by the way. Your sound is good.
Okay. Oh wow! I need to reconnect to you. Is it possible?
You need to reconnect. We see you.
Reconnect. Yeah. But still, it doesn't permit me to. Okay, I'll be in a second.
Well, I tell you what. Actually, how about if we swap order? Is Ashley? Are you ready? We could do Ashley next, and then Dmitry. Ashley, are you available? Thank you for being flexible. So we'll jump over to the next speaker, which is Ashley Gerlings, who will be presenting her abstract. Peri-transplant alemtuzumab levels predict risk of secondary graft failure and inversely impact CXCL9 levels after reduced intensity conditioning, HCT. And this is a correlative biology study to BMT CTN 1204 Ritchie.
Great. Can you hear me, Mike?
Yes, hear you great. Thank you.
Awesome. Okay, yes. So my name is Ashley Gerlings, and as Mike just said, I'll be presenting this research today. I won't repeat the title, but we'll just jump into the Ritchie study. So the BMT Clinical Trials Network 1204 Ritchie trial was a study that was conducted, and what Ritchie stood for was reduced intensity conditioning for hemophagocytic syndromes or select primary immune deficiencies. This was a multi-center, prospective, single-arm phase 2 trial of reduced intensity conditioning with melphalan, fludarabine, and intermediate timed alemtuzumab, and it was for patients with HLH and other PIDs. You can see the other PIDs listed here, and I'm showing you the conditioning regimen down here for those that aren't familiar with it. This conditioning regimen was picked based on retrospective single center studies that had showed improved survival compared to myeloablative conditioning regimens. And so the Ritchie study was able to enroll 47 patients at 22 sites between 2013 and 2015. And so here are the outcomes. This has been published in 2018. And so what the Ritchie study showed was there was good overall survival with a 1-year overall survival of 80%. And you can see it was similar for patients with HLH and other PID. The problem was there was a poor event-free survival due to graft failure. And so you can see here, the 1-year EFS was 39%. And the events that were included were donor chimerism less than 5%, or the need for a donor lymphocyte infusion or second transplant with or without conditioning. And the majority of these events were secondary graft failure. So why do we see this increased incidence of secondary graft failure in this patient population isn't known. One possibility is due to increased inflammation. And so Locatelli and colleagues in 2019 were able to show that there was an expansion of the recipient T cells and an increase in CXCL9, so the interferon gamma pathway, in patients who had early graft failure. However, in 2016, Marsh had showed that patients with high peritransplant alemtuzumab levels had an increased incidence of mixed chimerism and secondary graft failure. And this was likely due to an inhibition of the graft versus marrow effect. I'll just note that the transplant types in these two studies are different. In the Locatelli study, majority of these patients had alpha, beta, T cell depleted haploidentical transplants. Whereas in the Marsh study, it was mostly matched unrelated donors who had received bone marrow as their stem cell source without any T cell manipulation. And so our hypothesis was that this high rate of secondary graft failure in the Ritchie study is due to either an increase in inflammation due to the interferon gamma pathway or due to other cytokines, or it was due to high alemtuzumab levels. And so our goal was to evaluate cytokine patterns and alemtuzumab levels and their association with durable engraftment. So here's our methods. So obviously all patients needed to consent to be part of this correlative biology study. And then we were able to obtain stored samples from the BMT Clinical Trials Network. The samples had been collected on predefined days, as you can see listed here. For the cytokine analysis, there was over 100 different analytes that were looked at on from those specific samples on those predefined days. And they were looked at using the MEGS-PICS platform. To control for multiple analyses, there was a false discovery rate used. And then for the alemtuzumab levels, they were measured using a flow cytometry assay, and that assay has previously been published. So then we wanted to correlate these to our outcomes of interest. And so we'll just define what our outcomes of interest were. So first was primary graft failure. So that was defined as a donor chimerism less than 5% prior to day plus 42. Mixed chimerism was defined as a donor chimerism less than 95% on at least one occasion. And then secondary graft failure in this study is defined as a donor chimerism less than 5% after engraftment and or the need for a donor lymphocyte infusion or a second transplant with or without conditioning to manage mixed chimerism or graft loss. So now we'll jump into the results. So in this study, we had 33 patients. And you can see here, 75% of them had HLH, 67% were male, 82% were Caucasian, the 85% were pediatric. All of them received a marrow as their stem cell source. Majority had an unrelated donor, that was 82%, and 64% had an 8 out of 8 or 6 out of 6 match. As for our engraftment outcomes, what I'm showing you here in the second column is all the patients, but for those of you that are interested, we split them so you can see just the HLH and just the other PID patients as well. So there was one patient with primary graft failure. There was 22 patients who developed mixed chimerism, and so that was 67% of all patients. And of those 22, 11 patients went on to develop secondary graft failure, and that was 33% of all patients. And again, just reminding everyone that secondary graft failure included patients who had a DLI and or second transplant. And the outcomes in the HLH and other PID group were similar. And so now we are moving on to the cytokine analysis. And so as I mentioned before, over 100 different analytes were looked at and then compared in patients who had graft failure, which is represented by the green bars, and patients who didn't have graft failure, which is represented by the red bar. There were multiple cytokines that were noted to be different in these patient groups, but I'm going to focus on the CXCL9. So CXCL9 was different on day plus 14. And what you can see is patients who had graft failure, again, the green bar, had a lower level of CXCL9 compared to patients who did not experience graft failure. They had a higher day 14 CXCL9 level. Again, on day plus 28, we saw the same thing. Patients who had graft failure had a lower day plus 28 CXCL9 compared to patients who didn't experience graft failure. The one exception was the one patient who had primary graft failure, and that patient had a day plus 14 CXCL9 level that was significantly higher than the other patients who had secondary graft failure. So next, we wanted to relate the CXCL9 levels to secondary graft failure. And so this first analysis, what we did was split the patients based on the median day plus 14 CXCL9 level. And so patients who had a day plus 14 CXCL9 level that was less than or equal to the median, they had a higher incidence of secondary graft failure, as you can see here, compared to patients who had a day plus 14 CXCL9 level that was higher than the median. None of those patients developed secondary graft failure. And again, we did the same thing with day plus 28. If patients had a day plus 28 CXCL9 level that was greater than the median, none of those patients developed secondary graft failure. What I'm showing you here is just patients with HLH, which are green, or patients with just other PIDs, which is blue, so that you can see that the outcomes were these outcomes were similar, but when we looked at those patient populations separately. So our interim conclusion was that patients with secondary graft failure do not have evidence of increased interferon gamma pathway activity on day plus 14 and day plus 28 post-RIC HCT. So next, we wanted to figure out how this all related to alemtuzumab levels. And so what I'm showing you here is the alemtuzumab levels over the transplant course for all patients. But what we're going to focus on is what we call the day 0 alemtuzumab level. And that was calculated using the average of the day -1 and day +1 alemtuzumab levels. The median was 1.29 micrograms per mil, and the 25th percentile was 0.32 micrograms per mil. We decided moving forward to split our patients based on this 25th percentile, 0.32 micrograms per mil, because previous research has shown that the lytic threshold for alemtuzumab is around 0.1. And so this was felt to be a more clinically meaningful cutoff. So next, we split the patients based on that day 0 alemtuzumab level and looked at our outcomes of interest. Patients who had a day 0 alemtuzumab level that was less than 0.32, you can see here with the solid red line, that they had a lower incidence of mixed chimerism compared to patients who had a day 0 alemtuzumab level greater than 0.32. And you can see here the majority of those patients developed mixed chimerism. We did the same thing looking at secondary graft failure. So patients who had a day 0 alemtuzumab level less than 0.32, none of them developed secondary graft failure. Whereas patients who had a day 0 alemtuzumab level greater than 0.32, you can see a higher incidence of secondary graft failure. And again, I'm just showing you patients with just HLH, green, patients with just other PIDs, which is blue. We see the same results. So lastly, we wanted to correlate this with CXCL9 levels. And so again, we split the patients based on that day 0 alemtuzumab level of 0.32. And so the solid red line represents patients who had a day 0 alemtuzumab level less than or equal to 0.32. And you can see the CXCL9 levels are higher compared to the patients who had a day 0 alemtuzumab level that was greater than 0.32, which is this dotted red line. Their CXCL9 levels were lower. There were significant differences in the levels on multiple days, in particular on day plus 28 and day plus 42. We've added 2 boxes here to make this more clinically relevant. So the purple box represents CXCL9 levels that we see in healthy pediatric patients, whereas the pink box represents CXCL9 levels that we usually see in patients with untreated HLH. And again, you can see the patients with HLH, which is green, the other PIDs, which is blue, we see the same outcomes. And so that leads us to our conclusions. So we were able to show in this study that there's a strong relationship between alemtuzumab levels and durable engraftment, and that these alemtuzumab levels inversely correlate with CXCL9 levels. And so what we think is going on is that when you have a lower day 0 alemtuzumab level, there are some donor T cells that are able to persist. These donor T cells then react with the host hematopoietic stem cells, and this increases CXCL9. This graft versus marrow effect is what leads to durable engraftment. However, when you have a higher day 0 alemtuzumab level, there are less of those donor T cells around. So you don't have that graft versus marrow effect. And as a result, you're at an increased incidence of developing secondary graft failure. So moving forward, precision alemtuzumab dosing targeting a specific day 0 level will be important. However, it will also be important as we decrease those alemtuzumab levels to find ways to kind of deal with the inflammation as well. And then the other option would be increasing conditioning intensity, such as adding thiotepa. So I just wanted to thank the BMT Clinical Trials Network, as well as the PIDTC, all the centers that collaborated in this, and all the patients who contributed. Thank you.
All right, excellent. Thank you, Ashley. That was fascinating and a demonstration of what we're doing to our patients, I think, right? When we use our different conditioning regimens. So again, any questions, raise a hand. I thought I saw a hand that went away, so maybe not. Let's see here. So yeah, so I know these data, so I don't have any more questions.
Sorry.
Let's see here. All right, I'll give it another minute here in case there's any hand. Oh, here comes a hand. Oh, Karen. Go ahead, Karen, ask your question.
Yeah, I would like to know if there is a correlation between low donor T cells, high alemtuzumab level, and GVHD, or the other way around. So do you see more GVHD in those patients who have the low, the higher donor T cell levels?
Yeah, that's a great question. So the GVHD story is a bit complicated because you can obviously get GVHD when you have the lower alemtuzumab levels, but also if you're giving DLIs and decreasing your immune suppression, your GVHD prophylaxis quickly to manage mixed chimerism or graft loss, those patients are also at risk of getting GVHD. So when we talk about GVHD in this patient population, we kind of have to figure out, like, did they get the GVHD early or did they get it after DLI? And so if you look at patients who had a lower alemtuzumab level and excluded those that had it after DLI, there was, they do get more GVHD. But if you just look at the whole patient population together, the risk, the likelihood of acute, like grade 2 to grade 4 acute GVHD is actually the same between both groups.
Okay. If that answered your question, Karen, we'll move on to Ed, who also has his hand raised.
Sorry. Okay. Yeah. So since the day 0 measurement is really a surrogate for day -1 and day +1, do you think it's like too high of a day 1 or too low of a day -1, or vice versa, right? That's actually contributing to the effect that you're seeing.
I think it's how much alemtuzumab you have when your stem cells go in. So it depends, when did they actually get their stem cell infusion? Was it on day 0 or was it on day +1? And so I think the most important number is going to be when those stem cells kind of come in. And I think probably both those days are important because they are on the same trajectory. But I guess if you were asking me if it's more important to have I think, okay, going back, sorry, if you don't have if you don't have your stem cells in on day -1, then it doesn't really matter. It's when you get your stem cell infusion. So then I guess day +1 would be more important.
All right. Okay. Thank you very much, Ashley. So we'll move on to our next speaker. Dimitri, we have you back on.
Yeah, I'm ready.
I see a slide, so we hear you fine. So why don't you go ahead and proceed?
Okay, thank you very much, dear colleagues. Thank you for the opportunity to share the interim results of a multi-center study of safety and efficacy of vemurafenib and cytarabine plus cladribine therapy in high-risk pediatric lung cell histiocytosis with BRAF V600T mutation. So the current state of the art in high-risk pediatric LCH is well known. We know that cytarabine and cladribine are potent curative agents, but their use is associated with high toxicity, and vemurafenib is quite effective, but their withdrawal results in prompt relapse. What we've also recently learned that even in unifocal or multifocal LCH without risk organs lesions, we still can find the BRAF V600T mutation in myeloid precursors, and that's an update for misguided myeloid precursor theory from the Dutch group. We had some previous experience with vemurafenib, which was relatively good, but the problem was the same. The successful cessation of vemurafenib was possible only in 1 patient out of 5. So we decided to start a prospective trial for a combination of vemurafenib, cytarabine, and cladribine in children with LCH and BRAF V600E mutation. And we enrolled our first patient in June 2018. So here are the eligibility criteria: verified diagnosis of LCH. ERO+ patients could be enrolled at any line of therapy, and ERO- patients could be enrolled at the second of further relapse. BRAF V600 positivity, no documented heart disease, and informed consent sign. So we decided to combine the potential curative ability of cytarabine, cladribine, and the efficacy of vemurafenib. And here is the scheme of the protocol. So we start with vemurafenib induction, and later on we follow with 3 courses of cytarabine and cladribine. The doses are on the left. After the 4th course of vemurafenib, we continue with maintenance therapy with monocladribine, and after the 3rd one, we completely cease the therapy, and no other maintenance therapy was planned. So here are the endpoints to analyze: the overall response rate proportion at vemurafenib cessation, the progression-free survival from the therapy start, and what is more important, from vemurafenib cessation, the proportion of patients with severe adverse effects, and overall survival. So here are our MRD methods. We did ddPCR both in peripheral blood plasma and cell-free DNA, and we also derived myeloid progenitors from the bone marrow and did PCR in them. So here is our group characteristics. We had 2 groups, ERO+, ERO-. They had different age at manifestation, different age at protocol start, which is obvious. ERO+ patients were at different lines of therapy, and ERO- patients were at the 3rd or further line of therapy. There was a difference in initial disease activity score, and which is also obvious, but I'd like to mention that ERO+ patients were really heavily sick, and all patients completed the protocol. And here we have 1 patient lost to follow-up. We know she's alive and that she ceased her vemurafenib therapy, but we do not know her disease status. So here's the disease activity score pattern on vemurafenib induction therapy, which is completely in line with all other studies on vemurafenib. So vemurafenib induction was quite effective, which correlated with the biological nature of the disease. We can see the decrease in cell-free DNA BRAF V600T load. We also had some fluctuations even after the end of the therapy, but we think they're more or less insignificant. And we also can see the 1 patient who relapsed in risk organs, the only one. So here is an MRD load characteristic. So initially 2 groups were different in cell-free DNA BRAF V600T load and in myeloid precursors BRAF V600T load, but at vemurafenib cessation, they were more or less the same. So I think this is the main slide, the event-free survival probability by group. So we can see that ERO+ event-free survival was about 74%. So we had 3 relapses with median follow-up about a year. And ERO- event-free survival probability was about 80%. We had 1 relapse and median follow-up more than 1.5 years. And we can see that median time to relapse was 2.8 months, which is in line with other studies on vemurafenib. So the toxicity was quite mild. No vemurafenib-associated toxicity grade 2 or more was observed. And the mean period of neutropenia after cytarabine cladribine course was 8.2 days. And that probably explains the relatively low number of febrile neutropenias after the course. And no documented sepsis was observed. So this one is a busy table, but I'll try to explain it briefly. So we can see the BRAF V600T load in myeloid precursors initially at vemurafenib cessation and at the end of the therapy. So we can see that in patients who relapse in other sites from ear risk organs, relatively low numbers of BRAF V600T low T and myeloid precursors was observed. They are just the same as in patients who didn't experience the relapse. And in the only patient who relapsed with risk organs, we can observe the highest number of BRAF V600T low and myeloid precursors. And it was also the highest at the end of the therapy. Oh, excuse me. And this patient relapsed soon after the end of the therapy. So considering the relapsed patients, we continued mono-vemurafenib treatment on them, and we also watched closely the levels of cell-free DNA, BRAF V600T load, and myeloid progenitors BRAF V600T load. So, and if they were, if they reached 0 levels, so we tried to cease vemurafenib, and we did that in 2 patients. We did that successfully with EFS 21 and 6 months respectively. So as a conclusion, I'd like to say that the combination of vemurafenib and cytarabine plus cladribine is safe. Concomitant cytarabine and cladribine can be effective enough to cease vemurafenib therapy without a relapse. And myeloid precursors BRAF V600T load may predict the risk of relapse better than cell-free DNA load, but further studies should be conducted to prove that. And I'd like to thank everyone who contributed to our study, and especially my special thanks go to Michael Maschan, Alexei Maschan, and Daria Osipova. The study wouldn't be feasible without them. And I'd also like to thank our patients' families for their trust. And if you have any additional questions, I'd be happy to answer them.
All right, excellent. Thank you, Dimitri. That was fascinating. I have a question while we're waiting for people to raise their hands. Would you mind showing your schema slide of the treatment again?
Yeah, sure.
So it looked like, during the treatment, the patients received vemurafenib. Yeah, that previous slide. Yeah, with the one that shows the therapies.
Yeah, I'm trying to, this one.
Yeah, all right, yeah. So you actually did not administer vemurafenib for about 4 days or so. Is that correct?
Yes, 5 days.
Oops.
So during the chemo, vemurafenib was posed. Yeah.
Got it. And so no washout. In other words, they were on it, they got into their chemo, and they started right back on afterwards.
Yeah. On
okay.
The day +1.
Yeah. And was that just practical because you're worried about them being able to actually take it, or was that a sort of a rational feature here?
Both, because they could experience some emetic syndrome, and also we feared some interactions, and
Right. Yeah, because to me it's an interesting question whether or not vemurafenib might interfere with the chemo potentially, right? If you're blocking the active allele, then the cells are no different than wild type cells in essence, right? Potentially.
Yeah, that's the question, but we didn't know the answer actually.
Right, but there's no data so far other than yours. Yeah. All right, so it looks like Core has his hand raised. So Core, do you want to go ahead and unmute and ask your question?
That's quite a simple question. Nice presentation, Dimitri, but how did you get the vemurafenib? I would not be allowed to prescribe it this way in the Netherlands anyhow. So did you get it from the pharmaceutical industry, or did you just buy it?
A charity foundation helped us, Gift of Life Charity Foundation, bought that for our patients. Yeah.
Thanks. Makes it a lot clearer. Nice presentation.
Yeah.
Thank you.
And then also the schema, the Ara-C and the cladribine. So it's been a long time since we've used that regimen. This is a dose reduced from what was originally reported, by the French group. But as you mentioned, this was very well tolerated. That they're, I'm just kind of trying to wrap my head around it. That Ara-C dose is not really very high. But is this a sort of a regimen you guys have been using in other contexts, or this was specific to the trial?
No, that was, thank you for the question. That was quite a specific regimen, but well, we were a bit inspired with the Argentinian group regimen that was a bit mix of this, mix of that. So this is just made for this study completely.
Mm-hmm. Got it. Okay. All right. Jorge, you had your hand up a second ago. Did you have a question?
I would like to know, maybe I did not understand, could you give at the same time both treatments? I mean, chemotherapy and target therapy at the same time. It is possible.
Thank you very much for the question. I don't know. We did the pose for the 5 days of cytarabine and cladribine, but I think that maybe it is possible, but I don't know if it has the same efficacy, and if because of some certain problems like enigmatic syndrome and all this stuff. And also, we do not know the biological nature. So, would inhibition make the cells less susceptible to chemo? So that's an open question.
Okay. Thank you. Good presentation. Excellent.
All right. Okay. Thank you very much, Dimitri. So we're going to move on to our last presentation, and this is from Paul Kemps, who will, as he switches over, he'll be giving a talk to you on ALK-positive histiocytosis, the new clinico-pathologic spectrum highlighting neurologic involvement and responses to ALK inhibition. Okay, I see you there, down there, Paul.
Yeah, okay,
Right.
well, great. Well, hello, everyone. I would like to start by really thanking the organizing committee for allowing me to speak here today about our international retrospective study of ALK-related histiocytosis. So ALK-related histiocytosis is a distinct type of histiocytic neoplasm that was first reported as ALK-positive histiocytosis by Chan and others in 2008. And they reported it in 3 infants that had this multisystemic disease involving the liver, spleen, and hematopoietic system. And this included 1 case with a confirmed ALK rearrangement with a TPM3 ALK fusion. And interestingly, in all the 3 cases, the disease slowly resolved over many months. And in one of them, they even did without any specific treatment, but only with supportive care. And then in the following years, single case reports and small case series, including this larger series of 10 patients, confirmed the rare but indefinite occurrence of this distinct histiocytic entity. And in addition, they demonstrated that the disease may also present in older children as well as in adults, and that it may actually present as single or multi-systemic disease. So it really became clear that there was a bigger picture about the disease, but then the full clinical pathologic and molecular landscape remains incompletely characterized. So therefore, we set out to perform a large international retrospective study of this distinct histiocytic entity that we wish to call ALK-related histiocytosis for reasons that I will further explain at the end of this presentation. And in this study that was initiated by members of the Rare Histiocytic Disorder Steering Committee, clinical information was collected using a standardized CRF that was provided by the treating physicians, and then central pathology review was performed by Jennifer Pekarsik and Jean-François Emu. And then criteria for a diagnosis of ALK-related histiocytosis and inclusion in the study were first that there should be histologic confirmation of a histiocytosis, and then secondly, the case should have molecular confirmation of an ALK rearrangement, or it should have that classic infantile systemic disease with diffuse ALK expression as originally described in 2008. So in the end, we could include 39 cases that came from hospitals throughout North America, Europe, and Australia. And it immediately became clear that we had these distinct clinical phenotypic groups. So we had patients with multisystem disease, which we call group 1, and included 16 patients. And then we had these patients with single system disease, which we call group 2, and included 23 patients. And then patients with multisystem disease could be further divided into patients with a classic infantile systemic disease presentation, which we call group 1A, and included 6 patients. And then we had these other patients that also had multisystem disease, but did not fit that classic infantile systemic disease presentation. And we call that group 1B, and that included 10 patients. And if you take a look at group 1B and group 2, you can see that many of those patients had neurologic involvement. And two other things that really stood out is that we saw a female predilection across all groups, and that the majority of patients in our cohort were children. So if we then take a deeper look into the patients from group 1A, we can see that these were actually very similar to the original 3 cases reported by Chan and others in 2008. So these were infants that were 0 days to 5 months old, and that presented with hepatomegaly, anemia, and thrombocytopenia. And then 2 out of the 6 cases in our cohort had spontaneous resolution of the disease with only supportive care, but 3 others received first-line chemotherapy, and all 3 of those cases had either progression or relapse of the disease. And then luckily 2 out of the 3 cases obtained a complete response after second-line conventional systemic therapy, but also 1 of the 3 infants died from coagulopathy and sepsis. And this was a male infant that was 1 month old at presentation and had a KIT5B ALK fusion, and then had progressive disease despite 2 lines of chemotherapy, and then died at the age of 11 weeks. So this really shows the life-threatening condition that the disease can still represent, also in these cases, but that classic infantile systemic disease presentation. And then the final case was unfortunately lost to follow-up. So if we then take a deeper look into the patients from group 1B, we can see that these were 5 children and 5 adults, and included 3 infants without hematopoietic involvement. And the most commonly involved organs in the patients from this group were the nervous system, the bones, the lungs, the liver, the skin, and the lymph nodes. And the neurologic involvement included these pericardial brain masses, as is shown on the left for 2 children in this group, but also leptomeningeal enhancement or peripheral nerve lesions. Bone involvement really included lesions of the long bones in 7 out of 7 patients. And these were often visible as these hypermetabolic bone lesions on PET-CT imaging. And then finally, lung involvement was really uniformly characterized by these pulmonary nodules that range from micronodules to really significant tumors, as is shown here on 3 images of 3 children in this group. So if we then take a look at the treatment and outcomes of the patients in this group that had a median follow-up of 2 years, we can see that 6 out of 10 patients received conventional systemic therapy as first-line therapy. So that's either chemotherapy, steroids, or pegylated interferon. And then objective responses were observed in 3 out of 6 patients. So that's either complete or partial response. And then the other 3 patients that had either stable or progressive disease all received second-line therapy with ALK inhibition, and then objective responses were seen in 3 out of those 3 patients. And then 4 patients received first-line therapy with ALK inhibition, and objective responses were seen in 4 out of 4 patients, and the responses to ALK inhibition were durable in all 7 patients in this group that received ALK inhibition. So none of those patients developed progression or relapse of disease during treatment. So for example, here we can see some MRI images of 2 children in this group that received ALK inhibition. So on the left, you can see a child with these pericardial brain masses, and that received second-line therapy with lorlatinib and had this reduced response after 3.5 months of treatment. And then on the right, you can see another child that also had these pericardial brain masses, and this patient received first-line therapy with crizotinib and had already a reduced response after 3 weeks of treatment and a complete response after 20 months of treatment. So then finally, we can also take a deeper look into the patients of group 2, so the patients with single system disease. And these were 20 children and 3 adults, and included 12 patients with neurologic involvement, such as this child with a left trigeminal nerve tumor, and this child that had an intradural extramedullary tumor. And then we had...
Sorry, I'm still in conference.
And then we had 4 cases with single skin lesions, and 4 other cases with the soft tissue masses in various locations, like this child that had a single soft tissue tumor in the left lower leg. And then finally, we had 2 cases with unifocal bone lesions, often with really significant soft tissue extension. And then we had 1 case with a large single lung tumor. So if we then take a look at the treatment and outcomes of the patients in this group that had a median follow-up of 14 months, we see that 14 out of 23 patients received local therapy as first-line therapy. So that's either surgical resection or radiotherapy, and this was often effective. And then 4 patients received conventional systemic therapy as first-line therapy. 1 patient received first-line ALK inhibition, And the child with the soft tissue tumor in the left lower leg was observed without treatment, and then had a partial resolution of the tumor at last follow-up. And then we can also take a look at the second and third line treatments in this group. So 2 patients received conventional systemic therapy as second or third line therapy. And in none of those patients, an objective response was observed. And then 3 patients received ALK inhibition as second or third line therapy. And all 3 of those patients had an objective response. So then we can also take a look at all 11 patients in our cohort that received ALK inhibition, either as first or second or third line therapy. And then we can see that all 11 patients obtained either a complete or partial response to treatment, and that all 11 patients remained on treatment at last follow-up with a median duration on treatment of 16 months. So this really demonstrates the efficacy of ALK inhibition for this distinct histiocytic entity. And then, of course, the next big question that remains for further investigation is how long these patients should be treated, as is the case for treatment with BRAF or MEK inhibitors. So if we then finish the clinical part and take a look at the most important histopathologic features, it is interesting that classic xanthogranuloma features, including foamy histiocytes or two-tone giant cells, were present in about 1/3 of patients. But that the majority displayed a more densely cellular monomorphic appearance without foamy histiocytes, but sometimes more spindled or epithelioid morphology. So for pathologists, it is important to perform ALK staining in these cases. And then if they perform ALK staining, they have to beware of subtle ALK staining, because 4 cases in our cohort displayed focal weak ALK staining, and 3 other cases had this exclusive Golgi dot-like ALK staining, as is shown here in this image, which can be easily misinterpreted as negative. And therefore, we strongly believe that comprehensive molecular analysis should also be performed in ALK-negative histiocytosis cases when the detection of an ALK rearrangement could have treatment consequences. So if we then take a look at the molecular pathologic features, we can immediately see that the KIF5B ALK fusion was by far the most detected ALK fusion in our cohort. And then other ALK fusions were detected in 5 single cases. And then I'll have one final note before I end, and that is during this study, we also encountered 10 histiocytosis cases that had ALK expression, but did not have ALK rearrangements by comprehensive molecular analysis. And for example, here you can see one BRAF mutated malignant histiocytosis that had ALK expression, but did not have an ALK rearrangement. And this is really the reason why we think that ALK-related histiocytosis is a more appropriate term for the distinct histiocytic entity. And we think that this distinction is important because ALK-positive histiocytosis without ALK rearrangements might not benefit from ALK inhibition. So to conclude, ALK-related histiocytosis is really a distinct histiocytic entity. For clinicians, it is important to remember that it may present as multi or single system disease, and that it has frequent neurologic involvement in patients from group 1B and group 2, including in unusual sites like the CSF or the peripheral nerves. And we demonstrate durable responses to ALK inhibition in 11 out of 11 patients in our cohort that have been treated with diverse ALK inhibitors. And then for pathologists, it is important to perform ALK staining of histiocytosis frequently, but to beware of weak or exclusive Golgi dot-like ALK staining, and to know that molecular confirmation of the ALK rearrangement is really essential, as this may directly affect disease classification and therapeutic decision-making. So then I would like to end by really thanking Jennifer Pekarsik, Eli Diamond, and Jean-François Emile for leading this team effort. They're all members of the Rare Histiocytic Disorder Steering Committee. And of course, I would like to thank all other collaborators for participating in this research that really, I think, shows the benefit of international collaboration in the research of rare histiocytosis. Thank you.
All right, thank you, Paul. That was a very impressive and wide-ranging collaboration. So kudos to pulling this together. So it looks like Jorge has a question. Jorge, do you want to ask your question?
Yes. Thank you for this so good and so important presentation. I am interested if you have information about the liver involvement. What kind of liver involvement did you find in this page, in those patients?
Yeah, so it's a good question. So all 6 patients in group 1A, so all those infants had hepatomegaly, And then in some of those cases, we also saw focal liver lesions. So these were often visible as hypoechogenic lesions on ultrasound imaging, and on MRI, these were often visible as hyperintense lesions. And some of them also had the contrast accumulation. The same was also true for the patients in group 1B, so the patients with multisystem disease with liver involvement. They also all had these focal liver lesions on imaging.
Did they have cholestasis, important cholestasis?
So no, so and no, so they did not have cholestasis.
Thank you.
Except for one case. Yeah.
Thank you, sir.
Okay, all right. Other questions? Okay, all right, good. Well, thank you, Paul. So that concludes our the Presidential Symposium.